Today, we will discuss the T cell receptor and T cell receptor complex and the importance of these receptors for the T cell activation and the importance of these receptors for the T cell maturation process. Okay, first I will consider the T cell receptor. Then I will talk about the T cell receptor complex, means the associated molecules, which are also called as co-receptors, how they help in the activation of T cells and act as co-stimulatory receptors for the T cells. First, about the T cell receptor, TCR. In short form, it is called as TCR. The T cell receptor is made up of two different chains, that is alpha and beta chain. Therefore, it is called as heterodimer. And the alpha and beta chain is held by one disulfide bond just above the membrane. Just like in the case of antibody, the T cell receptor also has a variable region in the amino terminal region of the protein chain and constant region in the carboxy terminal region of the protein chain. It has only two domains, variable region domain and constant region domain. One constant region domain and one variable region domain. Now, each of these domains would have approximately 65 to 70 amino acids in the loop. And there is a disulfide bond that facilitates the formation of the loop. Okay. And the variable region domain has the variation in amino acid sequences, just as in the case of antibody molecule. Here also, the amino acid sequences are varied as per the requirement of binding to the antigen, because T cell receptor will establish the contact with antigen, which is presented by the class 1 or class 2 MHC. Okay, so as far as CD4 T cell is concerned, the alpha beta receptor is predominant in the CD4 T cell, and the variable region domain having that variation in amino acid sequence is recombined with the constant region domain, just like in the case of antibody. Okay, and different variable region domains have the possibility to combine with one constant region domain, just like in the case of kappa chain of the antibody. Kappa light chain had only one constant region and several variable region gene segments, and any one of that gene segment can be selected for recombination with the constant region. So the first formed T cell receptor on the T cell, that is during the maturation process in the thymus, and that T cell receptor will establish the contact with the self antigen presented by the thymic epithelial cell. Particular T cell, after establishing the contact with the self antigen on the thymic epithelial cell, depending on what class 1 MHC or class 2 MHC was involved in presenting the antigen. And we have already discussed, if it is class 2 MHC, that T cell receptor which established the contact with class 2 MHC will positively get selected to become CD4 T cell. And if the T cell receptor that was first formed establishes the contact with class 2 MHC, then that positive selection will result in the formation of CD8 T cell. And that stimulus of formation of CD4 and CD8 T cells is also going to be assisted by these other two co-receptors, which are called as CD3 complex. The CD3 complex is made up of the gamma epsilon and epsilon delta, which are also heterodimers, gamma epsilon and epsilon delta, which is called as CD3 complex, which are co-receptors. They're also there already when the T cell has undergone maturation in the thymus. Now, what is the importance of these CD3 receptors? I will talk about it little later. For the time being, we'll concentrate on TCR. Now, once the selection of CD4 and CD8 T cell takes place, that CD4 and CD8 T cell should now switch on the gene and recombination process to get the best fit receptor on its surface, just like the way B lymphocyte did. And there will be series of recombinations that will be taking place between variable region gene and constant region gene in the proliferating T lymphocyte. And I had mentioned to you that that receptor which is formed as the successive receptors after the proliferation, if it comes in contact with the self antigen, that T cell has to undergo apoptosis. But the receptor that is formed after the selection, if it puts in a best fit receptor for an antigen of the foreign origin, that T is going to be selected. And this process, we had spoken about it as negative selection. And majority of the amino acid sequences that are present here in the T cell receptors and the domains that are present there resembling the immunoglobulin domain. Therefore, this receptor also is called as immunoglobulin superfamily because it has some resemblances to the antibody molecule. And the variable region domain of the TCR resembles the FAB region of the antibody, fragment of antigen binding, because it establishes the contact with the antigen that is presented on the class 1 or class 2 MHC. Now, coming to another type of receptor, it could be instead of alpha beta, gamma delta receptor. Predominantly in CD4 T cells, it is always alpha beta. Once in a while, it may be in the process of selection of the receptor, it may be gamma delta. And that T cell will not be CD4 T cell, it will be either gamma delta T cell or it could be CD8 T cell. The CD8 T cell has the possibility of having the receptor of alpha beta type or gamma delta type, but yet it will be still CD8 T cell. And those cells which are helpful in the innate immune response, which become gamma delta T cells, they will retain gamma delta receptor as the major TCR, T cell receptor. Okay. Now, what is the speciality of this NH2 domain, that is the variable region domain? Just like in the case of antibody, it also has hypervariable region, which is called as CDRs, complementarity determining regions. And this also has three CDRs, CDR1, CDR2, CDR3. And these three CDRs, hypervariable regions in the variable region domain of the TCR, facilitates the interaction of that region with the best fit approach to the foreign antigen presented on class 2 MHC. Just for your information, I will show you the diagram once again for you to recall how it looks. Look here. This is the class 2 MHC that is presenting the antigen in its groove, and this is the major TCR which is having alpha beta heterodimer, which is establishing the contact with the class 2 MHC where the antigen is presented in the groove. And that makes the T cell receptor to receive the signal of that antigen, and this region of the TCR, that NH2 domain of the TCR, which opens out like a trough, it will be facilitating that T cell receptor to interact with the antigen properly and receive that antigen signal. and that will be fitting into the CDR1, CDR2, CDR3. And once that happens, that establishment of the contact takes place, the signal transduction should take place through the COH domain, which is present in the cytoplasmic region. The COH domain has a very short amino acid chain, that is about 5 to 12 amino acids. Because of the short chain of the domain, this receptor TCR by itself is not capable of stimulating the T cell, and that signal 1 that is going to be initiated by the TCR should need some assistance, and that assistance comes from these CD3 receptors, that is co-receptors. Therefore, always for the T cell activation, it is the T cell receptor complex that brings about the activation. And remember, when I had mentioned to you about the B cell receptor, which can activate the B cell only when alpha beta IgG domains, which are adjacent to IgM molecule, are also involved. Otherwise, just plain membrane bound single monomer of the IgM molecule is not able to activate the B cell. Why? Because the cytoplasmic domain of that receptor is very, very short. So that is the reason the co-receptors here also in the T cell, that is gamma epsilon and gamma epsilon delta, which are there as CD3 receptors, facilitate the activation of T cell. That becomes the signal 1. So in the membrane, transmembrane region, it is predominated by positively charged amino acids. Therefore, it is represented by positive charge. And there are about 21 to 22 amino acids in the transmembrane domain. And the cytoplasmic domain, I already told you, is very short. So therefore, the T cell receptor, if it has to activate the T cell, it needs the assistance of the co-receptors. And the co-receptors which are adjacent to it are belonging to the another complex called as CD3 complex. And altogether they are called as TCR CD3 complex. Now, TCR CD3 complex, which is present there, all of them will be involved in receiving the signal of the antigen. The difference between the CD3 complex and the TCR is that the gamma epsilon and epsilon delta does not have the variable region domain separately. The domain that is present has the affinity for the antigen, that's all. And it has only one domain, unlike in the TCR where variable and constant region domains are different. It doesn't have two different domains. It has only one domain, and that domain has a loop. Once again, about 65 to 70 amino acids are involved there. And the difference here is in the transmembrane region, same number of amino acids may be there, like 21 to 22 amino acids will be there, but it is predominantly made up of negatively charged amino acids, like aspartic acid and glutamic acid. Okay, and the cytoplasmic domain is longer. It is longer than the cytoplasmic domain of the TCR. The cytoplasmic domain would contain about 40 amino acids. In addition, there is a specific region in the cytoplasmic COH domain of the CD3 receptors consisting of a single amino acid residue, which is called as ITAM. ITAM. ITAM stands for immunoreceptor associated tyrosine based motif. MOTIFF, motif. Immunoreceptor tyrosine based activator motif. Immunoreceptor tyrosine based activator motif, which is called as ITAM. And that item is responsible because it is made up of tyrosine amino acid residues. It undergoes phosphorylation during signal transduction. More the number of items, better will be the signal. Okay. Therefore, the TCR, if you take, it doesn't have item in the COH domain or cytoplasmic domain. Therefore, it can generate only weak signal transduction, which is assisted by the CD3 receptor complex, which has a single item molecules on both of them in the cytoplasmic domain, and the cytoplasmic domain consists of 40 amino acid residues. So therefore, the co-receptor complex facilitates the activation of the T cell by generating signal 1 together with TCR. The TCR is the major receptor that contacts the antigen on the class 2 MHC antigen group. And CD3 undergoes activation simultaneously when the TCR is activated, and the cytoplasmic domains undergo phosphorylation to bring about the signal transduction. Next, there is another small adjacent receptor here on the left hand side of the TCR. Just check. It is made up of a short amino acid residue in the extracellular domain, which is hardly about 9 amino acid residues. A short amino acid residues, and that 9 amino acid residue chain is held by disulfide linkage, whereas in the CD3 complex there is no disulfide linkage between the heterodimers. And that short chain amino acid residue is made up of the protein chain zeta zeta or zeta eta. Two different molecules can be involved. About 60% of the time it is zeta zeta, and about 40% of the time it is zeta eta. Then it is heterodimer. Majority of the time it is a homodimer, that is zeta zeta. And the transmembrane domain contains once again acidic amino acids, that is aspartic and glutamic acid. Compared to other cytoplasmic domains, that is of the TCR plus CD3 complex, the cytoplasmic domain of this zeta zeta or zeta eta is a long chain of amino acids. If it is zeta zeta, about 113 amino acids are present in the cytoplasmic domain. And if it is zeta eta, 155 amino acids are present. That is, in the zeta chain, it will be 113, and the eta chain, it will be 155. And it has got 3 ITAM molecules. Just now I mentioned about the function of the ITAM. That is tyrosine based activator motif. There are 3 tyrosine based activator motifs that are associated with the intracellular cytoplasmic domain of the zeta zeta or zeta eta. And that is also involved in the signal transduction. So altogether, T cell receptor consists of T cell receptor of its own of alpha beta or gamma delta together with T complex, which is made up of gamma epsilon and epsilon delta plus zeta zeta or zeta eta. That is also included in the CD3 complex. Altogether, these receptors are responsible for signal transduction 1. Okay, and the subsidiary receptors are also called as co-receptors, and they're also very important. Now, when the monoclonal antibody studies were carried out to see the importance of these receptors, okay, all of them are very, very important as far as activation of the T cell is concerned. If you block the NH2 domain of the TCR, the T cell activation does not take place because it will fail to establish the contact with class 1 and class 2 MHC. If you block NH2 domains of the gamma epsilon or epsilon delta, the signal transduction will be incomplete. The T cell activation will not be proper. Similarly, the zeta zeta, even though it has got a very short extracellular domain, even if you block that, the signal transduction will be incomplete. T cell activation is prevented. Therefore, to cure certain autoimmune disorders, the monoclonal antibodies have been developed to block the T cells so that the T cells will not be activating and will not be facilitating activation of autoimmune response by developing monoclonal antibodies against the NH2 domain of the TCR or NH2 domain of the gamma epsilon or epsilon delta. Okay, so we will see the other receptors T cells. Okay, in addition to TCR and CD3 complex, which other receptors are very essential for the T cell activation? We had come across the receptor called as the CD4 and CD8 receptor. Remember, CD4 and CD8 receptor. CD8 designates the T cell to T cytotoxic cell or CD8 T cell, whereas CD4 designates the T cell to CD4 T cell or TH cell or T helper cell. Once again, recalling your memory back, I had mentioned that the CD4 receptor of the TH cell should simultaneously contact the beta-2 domain of the class 2 MHC. The antigen presenting cell is presenting the antigen of the foreign origin on the antigen groove to the TCR. At the same time, remember, the CD4 receptor was also responsible for the positive selection of TH cell when the first precursor TH cell or T cell, sorry, precursor T cell came in contact with the thymic epithelial cell, and the thymic epithelial cell in the thymus was presenting the self antigen on the class 2 MHC, and the first precursor T cell that came in contact with the class 2 MHC with the self antigen had established the contact with the beta 2 domain of the class 2 MHC through its CD4 receptor. And the triple positive T lymphocyte, it had CD4, CD8 plus TCR plus CD3 complex, which it had, will now delete the CD8 receptor because it was selected for class 2 MHC. And similarly, I had mentioned that the CD8 receptor of the CD8 T cell, which established the contact with the alpha 3 domain of the class 1 MHC when the thymic epithelial cell presented the self antigen to it first in the thymus, and the TCR came in contact with that self antigen presented on class 1 MHC, selected the T cell, precursor T cell, to become CD8 T cell. Why? Because the CD8 receptor came in contact with the alpha 3 domain of class 1 MHC, and it lost the CD4 receptor. So the CD4 receptor as well as CD8 receptor, the first function of them is to positive selection of the T cells to make them into TH4 or CD4 T cell or CD8 T cell. And once that has taken place, that T lymphocyte, which has been designated to become the TH cell or CD8 T cell, will be responsible for getting activated against the foreign antigen. Now let us see the structure of the CD4 T cell receptor. The CD4 T cell receptor is made up of 4 immunoglobulin-like domains. See here, D1, D2, D3, D4. The four immunoglobulin-like domain of the CD4 receptor resemble the constant region domains of the antibody. And within each loop, once again, there will be 60 to 70 amino acid residues which form the loop because of the disulfide. It is a single protein chain. Please remember, it is a single protein chain thrown into folds, four folds or four loops because of the intra-chain disulfide bond. It has also got a NH2 domain which is exposed exteriorly and the COH domain which is in the intracellular region, that is in the cytoplasm. Okay, a CD4 receptor has a molecular weight of 54 kilodaltons. All right, and each of those domains which are folded will give stability, and they are also associated, that each of those domains are associated with the carbohydrate. Therefore, it is a glycoprotein. Okay, it is a glycoprotein. Carbohydrate is associated. And when you compare CD4 with CD8, CD8 is a heterodimer, once again made up of alpha and beta domain. Okay, it is made up of alpha and beta chain held together by a disulfide linkage just above the membrane, and towards the NH2 terminus, it has a single loop because the intra-chain disulfide bond, and that single loop is also consisting of about 60 to 70 amino acids. And there is a difference in the molecular weight of these two heterodimers. Alpha chain is of 30 kilodaltons and beta chain is about 38 kilodaltons. Okay, so there is only one single loop that is on the NH2 terminus of CD8. Okay, and transmembrane domain made up of about 20 to 22 amino acid residues, and cytoplasmic domain made up of 25 to 27 amino acid residues. The cytoplasmic domain made up of 25 to 27, and transmembrane domain is 20 to 22 amino acid residues. Okay, and each of those loops will have about 60 to 70 amino acids, and altogether the entire CD4 protein chain would contain somewhere about 300 amino acid residues. Similarly, the CD8, each of those chains like alpha and beta chains would contain about 300 amino acid residues. Now, the speciality of these two co-receptors of cells is they designate the T cells into CD4 and CD8 T cells because initially they help in positive selection of the T cells in the thymic environment, and because they establish the contact with the class 1 MHC molecule in the alpha 3 domain by the CD8 receptor, and class 2 MHC molecule in the beta 2 domain of the class 2 MHC by CD4 receptor. And since they have quite a long cytoplasmic chain made up of 25 to 27 amino acid residues, they facilitate the T cell activation by signal transduction through the cytoplasmic domain together with the TCR CD3 complex, and they also will be responsible for the signal 1. So 1 that is generated for the activation of T cell consists of the T cell receptor complex that includes the TCR, CD3 complex plus the CD4 in the TH cell. In the case of CD8 T cell, the T cell receptor complex of TCR, which is probably made up of gamma delta or alpha beta with CD3, and CD8 complex. All together, they generate signal 1. And without the involvement of CD4 or CD8 receptor, it is next to impossible for the T cell to, in fact, get selected. So, if these receptors by any chance due to genetic deficiency are absent, T cell maturation process will not be complete. And after the maturation of T cells, if there is an autoimmune response in the person, by developing the monoclonal antibodies and blocking the NH2 receptor of the CD8 T cell, one can block the cytotoxic T cell response, because this association of CD8 with the alpha 3 domain will be blocked. Because of that, signal 1 will be incomplete. Similarly, when you block CD4 receptor NH2 domain by the monoclonal antibody, it will fail to interact with the beta 2 domain of the class 2 MHC. Then the signal 1 will be incomplete, and that TH cell will not be able to initiate the immune response. So now you know the importance of all these co-receptors. Now, lastly, we will talk about B7 and CD28 receptor. Why and how it is important in the T cell response. So first I will take CD28 receptor with interaction with B7 receptor. The CD28 receptor is expressed by the T cells, irrespective of the CD4 or CD8 T cells. Both of them are supposed to express these two receptors. Okay, the CD28 receptor is always expressed first, because it is responsible for the activation of the TH cell, which generates signal 2 after the contact with the antigen presenting cell with the first conjugate or first synapse of TCR with the class 2 MHC. The second synapse will be established between B7 and CD28. And if this synapse is not established, the signal 2 is not going to be generated in the TH cell or CD8 T cell, then that particular T cell will not be active. These two receptors are very important. Now, in that case, the B7 is classified into two subclasses, B7.1 and B7.2. B7.1 generally is called as CD80. Just like the way we call CD28, this is called as CD80, B7.1. And that is expressed in large amount, okay, large amount, by the antigen presenting cell when it encounters a foreign antigen and processes the foreign antigen. Then only the B7 will be expressed in large numbers on the surface of the membrane. B7, which is B7.1, CD80, has strong affinity to CD28 expressed by the T helper cell or CD8 T cell. And once it establishes the contact with the CD28, it is responsible for the generation of signal 2, and that will facilitate that particular T cell to complete its cycle of proliferation and formation of clonal expansion. Means larger number of T cells recognizing the same antigen. Okay, then when the immunosuppression has to take place, another receptor is going to be expressed at the last stage of immune response to suppress the immune response, that is called as CTLA-4 on the TS. That CTLA-4 stands for actually cytotoxic T lymphocytic antigen, because it was first discovered on the CD8 T cells. Cytotoxic T lymphocytic antigen CTLA-4 expressed on the TH cell or CD8 T cell is responsible for the immunosuppression. The reason being, it will establish the contact. It should not be it should not be expressed in the beginning. If it is expressed in the beginning, much before CD28, that T cell is not going to undergo activation. Remember that. It is always expressed to suppress the T cell activation and suppress the immune response at the end stage of immune response, when T cell activation further is not required and the immune is not required because the antigen load has gone down. There is no more antigen in the body. When there is no more antigen in the body, there is no point in keeping the T cells active. And if you keep the T cells active, they will keep on secreting the cytokines. If they keep on secreting the cytokines, other immune cells will respond to it, including the B lymphocyte, and they will keep continuing production of the antibodies. All will be waste. In order to prevent wastage, T cells have put in a regulatory mechanism. And that regulatory mechanism is such that once the antigen load has gone down, they have to suppress themselves. And that suppression comes with the help of expression of another receptor, that is CTLA-4. And the CTLA-4, which is expressed at the end stage, will establish contact with another B7 receptor, which is different from the first receptor. The first receptor, we have called it as B7.1, which is CD80. Now the second receptor expressed will be B7.2, and that B7.2 is CD86. The first one is B7.1, which is CD80, responsible for contacting CD28 for the stimulation of the T cells to undergo activation. Whereas B7.2, that is CD86, which establishes contact with the CTLA-4, which is in fact made up of the suppressive signal. When once it establishes the contact with that, it will switch suppression, and that interaction is 100-fold stronger than the interaction of C7.1. And because of that interaction, it will produce a strong signal inside the T cell or say T cell to show the suppression of the immune activity. Those T cells which express CTLA-4 finally have to undergo apoptosis. That is, they will no more show any activity. They have to be suppressed, and they will suppress all the gene actions of the cytokine production. They will suppress all the gene actions concerned with the T cell proliferation, and such a T cell now goes for apoptosis, means death. That is programmed cell death. Therefore, I said that CTLA-4 is expressed as a regulatory measure to control T cell activation at the end of the immune response only when the antigen load in the body has gone completely zero. As long as antigen load is there, the CTLA-4 is not going to be expressed. Once the antigen load has gone down, antigen presenting cells will not be encountering the antigen and they will not be activated anymore. And in such a condition, the CTLA-4 will be expressed and the immunosuppression takes place. Whereas CD28 is responsible for immune activation of the TH cells using that in interaction with the B7.1, it generates signal 2 to keep the T cell active all throughout and produce clones of T cell and activate the genes that are responsible for the secretion of the cytokines and keep the response going on, and it may help in even boosting the immune response in the immune system. So that is as far as the T cell receptors are concerned. Okay, and I will stop here. Thank you very much.